Hello students, welcome to a new class. Today we are discuss about the next part of the plasma membrane that is present inside the cell. Now in the last class we have discussed about what is plasma membrane, how it is act or how it is important for a cell for presence of the plasma membrane. We have discussed about the structure of the plasma membrane. We have discussed about different kinds of molecules that are present inside the plasma membrane and which actually save the cell from the external environment. We have also discussed in the previous class that different types of molecule can move inside the cell or can move outside the cell across the plasma membrane, and we have discussed about different types of movement, molecular movement, which can be present inside the cell for being alive or for life. Now today we have discussed about an example of special types of movement that can be present in a cell, that is called osmosis. In the last class we have discussed that in case of osmosis, this is actually the movement of water molecule across a selectively permeable membrane, and here plasma membrane act as the selectively permeable membrane for the osmosis, and it also allow the osmosis or allow the movement of the water for their requirement of the cell. Now here we are going to discuss about the example or how osmosis work, or how we can understand that osmosis takes place inside the cell in a special condition. If we provide the condition to the cell, how osmosis is possible and is there osmosis taking place or not. So in that case, first scenario we have to take an example that we take an animal cell, and salt solution. As you know that osmosis is only possible inside for liquid medium only. So in this experiment or in this experimental setup or in this example, we make a salt solution and use an animal cell for the showing of osmosis, and how, and we can discuss and we will see how osmosis takes place in the example. Now in the salt solution can be of three types. First of all, know any solution can be of three types. Type number one, it can be of hypotonic solution, it can be isotonic solution, and it can be hypertonic solution. So we need to know how animal cell can respond in three types of solution. So first let's talk about the hypotonic solution. First thing we need to know that what is osmosis. Osmosis is the movement of water across a membrane or the permeable membrane from higher concentration to a lower concentration. First thing in this type of solution, that is hypotonic solution, we need to establish that these characters are satisfying or not. That means we have a animal cell. Now we need to show that if animal cell, the plasma membrane of the cell can act as a permeable membrane and water molecule can move from higher concentration to lower concentration. Now how it is possible, how it is going on. In case of hypotonic solution, here first we make that here water is more and salt is less. As we have taken the salt solution, so here salt is granted for the amount of solute in this solution. So if water is more and salt is less, then in the surrounding medium what happens, concentration of water is more and concentration of salt is less. So in this case, concentration of water is more, from which concentration of, so if we think about inside the cell, then inside the cell there is less amount of concentration or the less concentration of water is present. So here in case of hypotonic solution, as the water concentration is more outside the cell and water concentration is less inside the cell, so in that case water move from outside to inside, because in the surrounding medium more water concentration is present, less solute concentration is present, or less salt concentration is present. So water molecule move from higher concentration to lower concentration across the plasma membranes. Here plasma membrane is the selectively permeable membrane, act as a barrier in the case of osmosis. Now in case of isotonic solution, here we can mark as water and salt are more or less equal amount. Water and salt, that is the concentration of water and concentration of salt, are more or less equal in amount, and the cell that is placed inside this isotonic solution, we can mark that the concentration of water that is present inside the cell and the concentration of water that is present outside the cell, that is the surrounding medium, both have the same concentration. So if inside the cell and outside the cell both have the same concentration, then no net movement of water is possible in case of isotonic solution. So we can mark this no net movement of water that is possible in case of isotonic solution. Now let's talk about the hypertonic solution. In that case of hypertonic solution, it is just opposite of the hypotonic. So in case of hypertonic solution, here water is less and salt is more. Water is less and salt is more. So in that case, if we place the animal cell in this hypertonic solution, then in the surrounding medium there is less water concentration, inside the cell there is more water concentration. We make the experimental setup just like that. Here surrounding medium, in that areas there is less water concentration, and inside the cell there is more water concentration. So what we know that in case of osmosis, water can move across a membrane from higher concentration to lower concentration. So in that case, water move out of the cell because inside the cell there is more water concentration, outside the medium there is less water concentration. So in that case, in the hypertonic solution, the cell become swell up. In case of isotonic solution, cell become as it is, and in case of hypertonic solution, cell become shrink. Okay, so these are the three different conditions of the cell if we placed an animal cell or plant cell in three different types of solution according to the water concentration, because in case of osmosis we can consider only the water concentration and water movement. So according to the condition of the solution, cell can be of three different type. In case of hypotonic solution, cell becomes swell up and can burst. In case of isotonic solution, cell is placed or cell is as it is just before the beginning, and in case of hypertonic solution, the cell become shrink. So these are the condition of the cell, or these are the condition of the osmosis according to the concentration of water inside the solution.